Hello, my name is Daniel T. Fuller, and today I'm here to talk about the seasonal taxonomic analysis of built environment viral bioaerosol data using Bayesian evolutionary modeling. So the built environment, or BE, airborne viromes are the collections of viruses found in and around human habitation. Um, the species that you find there, just like any animal species in any sort of biome, are going to vary by location, by season, and by the occupancy level of the other organisms, specifically humans here that are there. And a better understanding of these relationships on how viral diversity and how the viruses will relate to these can help prevent the spread of dangerous pathogens, especially when we're thinking about built environments such as airports and hospitals. Now, unfortunately, viromics lags behind other omics fields due to difficulties in sampling and difficulties in analysis. Here we have the built environment virome and what it's kind of made up of. Now, at the top here, we see known human-associated viruses, retroviruses, herpes viruses, coronaviruses, and the novel viruses such as SARS-CoV-2, the one that we're really interested in kind of looking at, right? That's what we want to know about. That's what we want to understand how these relate to our other variables. And along with these, you have plants, animals, fungus-associated viruses, and the bacteriophages related to the bacteria in the microbiome. There's a lot of stuff to filter through, right, to get at what we're really interested in. So this whole field is called metagenomics, where we're studying genetic material, and it has really exploded with the introduction of next-generation sequencing, where we can obtain massive amounts of data from these places in the built environment that we're interested in. Unlike bacteria and fungal data, there is no conserved marker gene, unfortunately, for analysis, so taxa comparison becomes difficult, and a lot of other analysis becomes difficult, and I will get into that shortly. But here's an example of some of the data we might be working with. This would be straight from an Illumina sampler or some other NGS sampler where we have, in the FASTQ format, we have quality reads, we have the actual sequences themselves, and so these have to be broken down, cleaned, trimmed, and then paired back together before being labeled to actually understand what we're looking at. Now, for our analysis of this virome data, what we're really interested in doing is relating season and occupancy to the built environment of viral diversity. Troubles going forward, right? We have the sampling difficulties for the actual viruses, which leads to sparse data, which leads to lots of noise because there's so many other viruses there. And then the difficulty with relating viruses taxonomically because of mutations and such things like that. We propose Bayesian analysis to solve these problems. It is stronger and provides stronger and more interpretable uncertainty quantification in terms of credibility versus confidence. Additionally, and very importantly here, an intelligent use of prior information can help mitigate that noise, and by playing with prior distributions, we can get a better understanding of what parts of the model, what parts of what the system we're working with are actually generating noise. Now, standard approaches for Bayesian analysis in metagenomics. You often see software for Bayesian phylogenetic analysis, such as Mr. Bayes, very good at what it does, but has limited functionality. One of the ways in which we can use it for this project, something like a gamma distributed GTR evolutionary model, where we are assuming transition probabilities are equal in both directions, and assuming that rate variation between sites follows gamma distribution. This is an example of a phylogenetic tree for a gene from conserved through mammalian species, and we see that we have very high credibility values for the tree that we've made. We're very confident in it, but if we were to apply this to viruses, and we have, we see that it is much less certain because the virus, comparing the whole sequences of viruses, there's a lot more differences than you have between a single gene between species, right? And so that makes it a lot more difficult in general to work with these. We would like to look at more than phylogenies, right? And so for this, we put forward STAN, Computational Engine Sampler for Bayesian Models. It's really top of the line in its field, and we are moving forward with a seasonal prediction model using viral diversity to predict the season, occupancy, other things like that. We can use gamma diversity or parameterize gamma diversity by alpha and beta diversity, as well as the presence of each species individually as dichotomous variables, and then using season as our predictor variable in a multinomial logistic regression model in a Bayesian style, as shown here. And this is really what we want to move forward with using and what we have been using testing on this data. But testing on data that we've obtained from NCBI really isn't quite as exciting as it will be when we obtain proper data with an efficient and replicable processing pipeline from the local hospital here, from the university, and from other areas. We are going to be obtaining our own data very soon that we will be able to apply this workflow through to, and we're very excited for that. Additionally to this, we can compare the results of the Bayesian seasonal predictive model we've made to standard machine learning force classifiers, such as those presented in Preston et al. 2019, which are more commonly used in the field at this point. Then reversing this model to predict the diversity metrics based on external factors and measures. Training this on our local data will be a great step towards our final goal of predicting specific types of viral outbreaks from changes in virome dynamics, which is where we can really make a difference with this project, and that's what we're really excited to do. To learn more about this, please enjoy the other presentations from our group, and thank you very much for your time.